Okay, hello everybody. Good evening. This is week 9 of these extra sessions for the particular course, Classics in Neuroscience. So till now, we have gone through multiple, basics for this particular subject, neuroscience. There were multiple foundations for various different subtopics that we have established. Uh, We'll first proceed with the previous year questions that were asked pertaining to week 9. We'll discuss those questions and then we'll switch to our ongoing discussion about learning and memory, right? So we'll see how biologically or molecularly the mechanism is in effect while we are potentiating memory, or even in case of our working memory, how explicit versus implicit memory is getting stored. So that discussion we were already having in our previous sessions. We'll continue with that discussion as well, right? So first let us begin with this quick previous year questions part, right? So it is fairly easy because it has more to do with history and less to do with concepts this particular week. So we'll quickly sift through it, right? So the first question that was asked was, who is considered the father of modern neuroscience? So this is something that we have discussed even in the beginning of our course, maybe in the first or second week itself, right? So the options that were given were Robert Barony, Camillo Golgi, Santiago Ramon y Cajal, and Sir Charles Sherrington. So which one of these is considered as the father of modern neuroscience? The correct answer for this is Santiago Ramon y Cajal, right? So he was a Spanish scientist who has even won a Nobel Prize for a concept which we are going to study. So I'm not naming it right now, right? And he has actually, in fact, shared that Nobel Prize with Camillo Golgi, and he is known as the father of modern neuroscience, right? So, what was Santiago Ramon y Cajal's major contribution to neuroscience, right? Was it the discovery of neuron doctrine, or development of the reticular theory, or invention of the microscope itself, or discovery of neurotransmitters, right? So actually, the discovery that he and Ramonica and Camillo Golgi together were given a Nobel Prize for is neuron doctrine. But he was the one who has actually given the neuron doctrine. The reticular theory, in fact, is what was proposed by Camillo Golgi, right, instead of him. So, in the neuron doctrine, for which both of them were given the Nobel Prize in Physiology or Medicine back in 1906, what Santiago Ramon y Cajal had proposed was the nervous system consists of single cells with no cytoplasmic continuity between these cells, right? So each of the neurons is separate. There is a network of these separate cells that is actually building up the neural system, is what he said. What Camillo Golgi, on the other hand, said was what was the part of the reticular theory, which is the nervous system is a network of finer branches that ultimately are continuous, like the vascular system, right? So this was later proven to be incorrect because, unlike vasculature, these are not continuous. There are small gaps in between, and there is a connectivity that is present through synapses, which are in fact gaps, right? So, like, it is not as connected as the vascular system itself, right? Which is the blood vessels in your body, right? So, in fact, yeah, as you can see, both of them gave contradicting opinions, right? So one said that it is continuous network in the brain. The other said that it is not a continuous network. It is a network, but it has breaks in between, and these are made. The network is made up of single cells rather than one single cell forming the entire network, right? Unlike what Camillo Golgi said. But even though they had contradicting opinions on them, their contribution to neuroscience was such that both of them were actually awarded the Nobel Prize. One other reason for that is again, we'll come to that shortly while discussing the questions itself, is because Camillo Golgi might have developed a technique that helped Santiago Ramon y Cajal to come to this conclusion that was eventually correct also, right? So that is why also both of them were actually awarded the Nobel Prize in medicine in 1906. So this is the technique that I was talking about. What technique did Camillo Golgi develop to visualize neurons, right? Was it the double impregnation method or the Black reaction or electron microscopy or neuroplasticity staining, right? So which one of these was developed by Camillo Golgi? Maybe you have not read up or we have not discussed this particular term, but we have discussed another term for this particular kind of staining, which is silver staining, as we have discussed it before. It is also known as black reaction because the silver basically deposits in the neurons and also particularly in the cell bodies of the neurons, leaving a dense dark black colored stain on these cell bodies, right? So that is why it is also known as black reaction, right? Often it is done with multiple silver salts. The most common one that we currently even still to till this date use in labs is silver nitrate staining, right? So nitrate salt is commonly used. So this is from one of our previous lectures. I have picked up a few slides to give a sort of a recap, right? So Camillo Golgi, this gentleman, developed a method of staining with silver salts that revealed their entire cell structure under the microscope. Right? So he could see clearly that each neuron typically has a cell body like this and two types of processes, the branching dendrites at one end like this and a long cable like axon at the other, right? So they had two kinds of processes. One was a long process and one was shorter processes. The shorter were more branched, long was less branched, right? So the more branched, shorter ones were dendrites. The less branched, longer one was axon. Then he, based on this staining and how he saw these neurons, gave the reticular theory. So if you see over here, it stated that the nervous system is composed of a single continuous network of cells rather than separate nerve cells, right? So this is definitely not true. We'll see shortly why it is not true. The theory was proposed by Joseph von Gerlach in 1871 and it was later popularized by Camillo Golgi after he stained the brain sections and saw these kinds of networks in the brain, right? Then coming to question number 4. What is the Baronet test used for, primarily used for? Right? So is it to test visual acuity or assessing vestibular function or to measure the heart rate or to examine brain plasticity? Right? So which one of this it is primarily used for? It is actually used for assessing the vestibular function. Now, what is vestibular function and what is vestibule? Right? So vestibule is a part of the inner ear actually, right? It is a part which has those semicircular canals. We have got when we were talking about the five senses, we have briefly talked about the structure of each of these senses. So I remember speaking about this inner ear, right? We have revised the structure of the ear itself, right? The outer ear has this pinna, earlobe, and the canal, right? The middle ear has those three bones, malleus, incus, stapes, right? Our hammer, anvil, stirrup, as we call it. And the inner ear has two different major structures. One is the oval window, which is attached to that those three network of the middle ear bones, right? So that is which is leading to cochlea. which has hair cells or the receptors that actually understand or grasp the frequency that is coming and send the signal to the brain, right? So there are mechanoreceptors that convert the signal to something that can be transferred to brain and understood by the brain. The other region is the semicircular canals that is present in the brain. That region is actually known as the vestibule. What these semicircular canals essentially do is they help to maintain the homeostasis and sort of balance in our body, right? So if we are sitting, if for instance I'm sitting upright, right, I am not drooping like this or drooping like that, even though gravity should be pulling me down, right? But like at least my head is upright, right? So there is something in my body that is actually at function which is maintaining it in this way, right? That I am not drooping about or I am not falling this way, being pulled by the gravity, right? So that is the function of that inner ear portion, vestibule, and the semicircular canals. So to assess that function, there was a test that was invented by Barney, which came to be known as Barney test, right? So, what is this Barney test? It is known as the caloric reflex test. It will help you in the next question, which evaluates the vestibular function by stimulating the inner ear to observe involuntary eye movements, which is also known as uh, nystagmus, right? Barony test is a diagnostic procedure developed by the Hungarian otologist. Otologist is somebody who has something to do with the ear, right? The doctor of the ear, specifically inner ear also, Robert Barone, in the early 20th century to assess the integrity of the vestibular system and the vestibulo-ocular reflex, which is known as VOR, which coordinates eye movements with head motion, right? So what does that mean? If, for instance, yeah, I move my head like this, right? I will if I intentionally want to, I will keep looking over here. But naturally, if I move my head over here, my eyes will also move over there. If I move my head over there, my eyes will also move over there, right? Naturally, if I do that instinctively, right? If I'm thinking about it, then I can keep my eyes over here and suppose move my head there. That is a different thing. So, it is commonly used to detect the vestibular disorders such as benign paroxysmal positional vertigo, uh, Meniere's disease, labyrinthitis, and vestibular neuritis, right? So these conditions, which are mainly conditions of or related to the inner ear. So this is kind of what happens in a Barony test. There is the chair that you are, bound to or you are made to sit on, and the chair is inclined. It can be done manually also. So if even if there is no chair, you are kind of blindfolded, and the doctor will tilt you at an angle, bring you back, and uh, uh, they'll basically test what kind of a reaction you are given, right? So it is used to identify the affected ear in unilateral vestibular disorders. Also, it is used to assess the severity and symmetry of vestibular function. Also, to monitor the effectiveness of vestibular rehabilitation or treatment after, say, a surgery or a procedure, right? In this, there is either air or water pressure that is put in your ear, and then the reaction of your VOR, vestibulo-ocular contact is tested after, tilting you here, there, so that it can be assessed whether that homeostasis is being maintained or not, right? What was the key finding of Robert Barone that led to his Nobel Prize? So, Robert Barone was also a Nobel Prize winner, right? Was it the law of dynamic polarization, or discovery of the caloric response, or neuron doctrine, or reciprocal innervation, right? So we have already, as I pointed out in the previous discussion, that it is going to help you in the next question. Discovery of caloric response or Barony test. which is actually used to discover this caloric response, was a key finding of Robert Baroni that led to his Nobel Prize, right? No, neuron doctrine we have already seen was shared by this Nobel Prize was shared by Camillo Golgi and Santiago Ramon y Cajal, right? Also, it is interesting to note that this Nobel Prize was solely given to Robert Baroni, not shared with anybody, right? So you can see prize share one by one. So solely awarded to him. He was awarded the Nobel Prize in Physiology or Medicine in 1914. Uh, he was born in Austria and he died in Sweden, right? Sweden is actually also the country that where the Nobel Prizes are given. So affiliation at the time of the award, when he was awarded the award, he was still at Vienna University in Austria. The motivation for his prize was for his work on physiology and pathology of vestibular apparatus, right? So he received his Nobel Prize 1 year later in 1950. So this is the science for which he was awarded his Nobel Prize. The inner parts of the ear play an important role in helping us maintain balance, as we recently talked about. Irritation of the inner ear causes vertigo and spasmodic eye movements, which is also known as nystagmus, right? So, vertigo is something that we have heard about, probably in people around us also at times. But even spasmodic eye movements are associated with these inner ear infections, right? Barony discovered that nystagmus occurred in one direction when he syringed a patient in ears with cold water, and in opposite direction when he injected warm water, right? The explanation was the changes in temperature made the fluid in inner ear canals rise and fall respectively. This discovery had a major impact on the treatment of diseases of the inner ear later, for which he was actually recognized via this Nobel Prize. Just give me a moment. All this irritation talks has reminded me that I am also having this an irritation in the eye, so I should wear my glasses. Yeah. Okay. It will help me not get a migraine later. Okay. Yeah. Moving on to the next question. What is the law of dynamic polarization proposed by Kahal? Right? So, does it say that the nerve impulses travel in both directions in the neurons, or that nerve impulses travel in one direction within the neurons, or synapses transmit electrical signals without gaps, or nerve impulses only travel through dendrites? Right? So, which of these is the correct answer? It is the second one. What he said, Kahal at that time is through the law of dynamic polarization is that the nerve impulses travel in one direction within the neurons, right? We also studied how this directionality is maintained because of the depolarization of the previous region. Uh, the nerve impulse only has an option to proceed in one direction, right? That is how this directionality is actually maintained. We have gone through the mechanism also in the earlier lectures. So, if you look at the read about the law of dynamic polarization, the dendrites generally receive the synaptic input. Uh, that is over here. This is the region of input. They are lying in the postsynaptic region for the previous neuron. That is what is mentioned over here. The signal is further integrated over here, and action potential. If it causes an action potential, it is generally initiated over here in the axon hillock. And then this is where the output is transferred to the next neuron, which will be the uh, 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 next in line, right? So axons are generally send the synaptic output, and this is the presynaptic terminal for the next neuron, right? So, what condition did Baronet's research on vestibular system help explain? Right? Was it Parkinson's disease or Alzheimer's disease or vertigo or epilepsy? So, we have just read about this also that it was vertigo. Yeah. Right? So, what are these other conditions? Both of the first two are neurodegenerative disorders. Parkinson's disease has something to do with the loss of dopamine secreting neurons or dopaminergic neurons. Alzheimer's disease has something to do with the loss of neurons in multiple regions, which is associated with alpha-synuclein or APP or tau aggregates in the brain, and epilepsy is a an increased hyperactivity, you know, phenotype of the brain, right? Wherein there is a hyperactive state of the brain, wherein the brain waves are electrically hyperactive, which is causing some kind of a dysfunction in the body or the brain. So, what is vertigo actually? You can see again, let us revise the structure of the inner ear first, right? So this is the ear, right? This is the outer ear. It has the earlobe and the ear canal, as we talked about. The middle ear, which has the eardrum, malleus, incus, and stapes, these three bones. And then the inner ear, which has a cochlea, which is responsible for the hearing portion, and semicircular canals, which is responsible for the balance, right? So you can see in the semicircular canal is the vestibular nerve also, and this is actually the vestibular region. So this inner ear, when we talk about, uh, is further, if you want to further label it, this is the cochlea that it is circled. It has a lining of hair, like hair cells in within the cochlea, which is receiving the frequency that is coming from outside. These semicircular canals have some fluid that is filled in it, and there is something known as utricule and saccule in it, right? In normal conditions, what happens is in the semicircular canal, there are nerve fibers, sensory hair cells, there are hair, gel membrane, and there are crystals which are known as autoconia that are arranged on top of this gel membrane, right? But over here, what happens is, in case of vertigo, which could be due to some primary infection, secondary infection, some kind of a defect in this anatomy or this physiology of the semicircular canal, uh, this top layer of the autoconia or the crystals, it is somehow shifted, right? Due to which, so these crystals can float into the wrong part of the canal also, right? Due to which, what happens is that the balance that they used to create is no longer maintained, right? So, what is vertigo? It is a sensation of spinning or tilting, or that one's surroundings are rotating, right? Why is this happening? It is basically happening because of shift in these particular crystals or these cells, right? So they should remain at one region, right? That is how they maintain balance. If they start going haywire, then your balance will also be lost, the balance that they are maintaining. So, vertigo is a common yet often misunderstood condition which affects many individuals worldwide. It is characterized by a sensation of dizziness or spinning, which is often accompanied by nausea and difficulty in maintaining balance. This vertigo is not a disease in itself, but rather a symptom of an underlying issue within the vestibular system, which controls our sense of balance as well as spatial orientation, right? So I hope this particular condition is also clear to you what exactly is vertigo, right? So who investigated the phenomena of vertigo? We have just read about him. We have seen how he has won a Nobel Prize also, and how that test which is done to understand whether there is vertigo or not is also named after that scientist, which is given over here. Oh no, that is actually given over here to maybe confuse you. I just remembered. Yeah. So, was it Ernst Mach or Crumb Brown or Perkinjay or Barony, right? So, the correct answer actually is Perkinjay, right? He was the first person who investigated the phenomena of vertigo, right? Barony was the one who further, investigated it. He gave certain insights as to what exactly is vertigo, and he also invented the test that is used to understand this, whether a person has vertigo or not, or how the vestibular system is affected, right? So, who investigated the function of the pigeon's brain? Was it Pierre Florence or Charles Sherrington or Cahal or Ernst Mach? Right? So, the correct answer over here is Pierre Florence. He was the one who investigated the functions of the pigeon's brain. What did Cahal's staining method signify? Did it support Golgi's reticular theory? So we have already studied that it did not. You can read the other three options. To identify firing rate of neurons, to distinguish between gray matter and white matter, or to identify the neuronal cell body. So the correct answer is to identify the neuronal cell body, because he did it in autopsy brain sections, right? Not in actual brain, in vivo or in a particular organism. Rate of firing he could not understand, but he could identify the various kinds of neuronal cell bodies, for instance, right? So that is also one of the reasons why the reticular theory was disproved, that it is not one continuous network of the same cell, because there are different kinds of neurons that are actually present, right? So Ramon Cajal, he actually used Golgi's technique, but he discovered that nervous tissue is not a syncytium or a continuous web of elements, but rather it is a network of discrete cells, as we have talked about before. So, in the course of this work, Ramoni Cajal developed some of the key concepts and much of the early evidence for neuron doctrine, for which he was awarded the Nobel Prize. The principle that individual neurons are elementary building blocks and signaling elements of the nervous system, right? So you can see over here, there are different kinds of neurons, right? This has a different kind of structure. This has a different kind of structure. This has a different kind of structure. So different kinds of neurons were present, and his drawings are actually compiled in this very, fascinating book, which is The Beautiful Brain, which has a copy of the hand drawings done by Ramon Cajal, right? So he was a scientist and an artist at the same time. And he has documented this art of what he was seeing through the microscope really nicely. So that is it for this particular part of the previous year questions. We'll switch back to our discussion of learning and memory now, right? So that is an ongoing discussion we are having. We'll revert back to that discussion. Yeah. Okay. Please give me a short moment before we actually begin. 1 second. Okay, thanks for that. I'll just reshare my screen. Okay, so we'll begin with our discussion about learning and memory then, right? Uh, okay, I wanted to keep a recap of this, which I have forgotten. Let me just open that other slide also. Yes, so we'll do it like this, right? So that we have that continuity, like I gave you a brief recap. I just went over what we had, talked about in the previous session last week also. Since it is, week three of talking about learning and memory, we'll not probably revise from week one, but we can revise for the previous week what we have done, and that then it'll be easy for us to, understand where we are picking it up from, right? So we had discussed in the week prior to that that there are two kinds of memory, implicit and explicit. Implicit is the kind of memory wherein you are not probably consciously thinking about something, suppose language, or when you are driving, you remember how to drive, or you remember how to ride a cycle. You are not consciously trying to remember how to ride a cycle or how to drive, right? So those kinds of memories are implicit memories, and there are other kinds of memories that we have to consciously recall, such as the name of a person, the birth date of a person, the phone number, or some information that you need to write down in an exam. So all that is explicit memory that you intentionally have to recall, right? So we had studied that implicit memory supports perceptual priming, right? Which means that what is this priming? Priming, it is a type of this implicit memory that operates in amnesic patients as well as healthy patients, wherein again, it is the same concept that we don't require conscious effort to, remember something, right? What is conceptual priming versus perceptual priming? Conceptual priming provides easier access to task relevant semantic knowledge because knowledge has been used before. It is correlated with decreased activity in left prefrontal regions that subserve initial retrieval of semantic knowledge, while perceptual priming occurs within a specific sensory modality. Um, right? So such as information about form and structure of words and objects. So, implicit is more perceptual than conceptual. We had gone through this example of chair and apple and how it gets easier to read it once you have done it before. And then we had also studied how implicit memories can further be of two types: associative or non-associative. Non-associative, in which we learn about properties of a single stimulus, while associative is when we learn to combine two unassociated stimuli, right? Stimuli that previously might not have had a relationship between them. So, when we talk about non-associative learning, there could be habituation, sensitization, and dishabituation that we talk about. As we, introduced this last time also. For instance, um, I am sitting over here and there is a blast like sound outside, right? I will be alarmed because there is a blast. Naturally, anybody will be alarmed. But at the same time, if I know that it is the Sahara and around that I hear a blast-like sound outside, I'll be habituated. I will not probably respond to it as much because I know that it is continuously going to be happening for a couple of days, and I know the cause of it. So I'll be a little desensitized or a little habituated to that fact, right? I'll not respond as much, right? On the contrary, what is sensitization? For instance, uh, you know, there is a certain phenomena to which I am conditioned, due to which I respond to it even more, right? For instance, if I burnt my hand uh, with hot water, right? So some really hot steaming water spilled on my hand and I burnt my hand, right? So I'll be very, very sensitized to it. So even if the water, whenever particularly I'm heating water or I'm going to use the water from the geyser, I'll be very, very sensitive to the kind of stimulus that I might receive. So that kind of a thing might be sensitization. So I am overly cautious about a stimulus, right? So it is almost opposite of habituation, right? And also there is something known as dishabituation, which is sensitizing stimulus can override the effects of habituation, right? For instance, I got habituated to that blast sound around the Sahara or Diwali. But again, a few months passed by, I had not heard that kind of a sound. I got dishabituated, and then again, that kind of a sound came, and I will respond more to it because I can get sensitized to it again, right? Then there were the associative experiments which included conditioning, which could either be classical conditioning or operant conditioning, right? Classical conditioning is that very famous landmark example of Pavlov, Ivan Pavlov, right? Who basically conditioned a dog to hear a bell ringing when there is food given to that dog, so that even when the food is not provided but the bell is rung, the dog will salivate because it is anticipating that the food will come. So it is associating this ringing of the bell to receiving of food, right? So that is why it will salivate. So this is what is classical conditioning, right? And then again, there is operant conditioning, wherein there was reinforcement that was done, that a particular kind of behavior might lead to a particular kind of result, right? If, for instance, the mouse presses the lever when the yellow light is shown, it will receive a shock. But if it presses the lever when the blue light is shown, it will receive the pellet. But how will it know this, right? Nobody is telling this to the dog, to the mouse, right? So it will have to try, via trial and error, it will have to learn this, right? So that is what is operant conditioning basically. Then there were two forms of long-term memory that we saw. One was implicit or non-declarative, and then it was explicit or declarative. Then there were various regions in the brain that were associated with each of these subtypes of these memories, right? So for priming, for instance, neocortex was responsible. Procedural striatum in the hippocampal region was responsible. Then associative learning, if it is related to emotional responses, then amygdala, which is that center for fear and emotion. Or if it is related to skeletal musculature, then cerebellum is responsible for that. Then the non-associative learning, such as habituation and sensitization, the reflex pathways might be involved with that, right? Similarly, for explicit, for both the semantic and episodic memory, which is related to either facts or events, the medial temporal lobe is responsible, right? Then we saw how these memories are actually stored, right? So there is some kind of a synaptic involvement in, involved in the effectiveness of storing of a memory. So habituation, for instance, results from activity-dependent presynaptic depression of the synaptic transmission, right? So these were examples that we studied. We also saw how a simple a model organism was used who had a simple nervous system with not so many neurons or not so many dendritic processes, so that these responses about habituation, sensitization, etc., can be studied more easily. Uh, And we also talked about long-term habituation and how this happens, right? It is basically caused by decrease in number of synaptic contacts between sensory and motor neurons. So, because habituation will lead to what? It will lead to a decreased response. So, less number of synapses, less amount of response, right? That is what we talked about. Then sensitization will be the opposite of habituation. It will have more number of synaptic, points that are involved over here. So that is what. Also, um, uh, okay, all this we have already discussed about. So I am not going to get into the details of it again, right? So this is what we were talking about. So long-term habituation, lesser synapses, long-term sensitization, more synapses, right? So that is what happened. So this was the final one that we discussed. And this was an actual image that was taken for an organism that proved that there could be long-term sensitization because of increased number of synapses over here, as it has been shown in this, right? So number of synaptic photons or N feed were increased over here. This was our discussion in the previous session, right? So now from this session onwards, we can begin our discussion, and we can talk about how, how we are actually learning fear, how amygdala, hippocampal are responsible for this learned memory. And further, we'll also discuss about how working memory is, getting, how that is happening, how where it is getting stored, and if it is getting stored. That is what we are going to understand, right? So the first statement over here is memory for learned fear in mammals involves amygdala. So we have already studied while we were looking at the different brain regions that what is amygdala responsible for? It is responsible for uh, emotion and fear, basically, right? So when we are learning to fear something, it is also probably getting, uh, you know, either recognized by or stored through amygdala, right? So there are two types of fears, right? One is the innate fear, which is the ability to recognize and respond to danger, and it is necessary for survival, right? So this comes from within us. We have not learned this fear. It will probably come from within us, right? The other is okay, because innate fear has been conserved throughout the evolution of species. One can readily discern and study fear in a variety of experimental animals, right? At the beginning of 20th century, both Pavlov and Freud independently discovered that fear can also be learned, right? So what is this learned fear? It prepares the individual for fight or flight if there is even the suggestion of an external danger. So even if you are not in danger as yet, but it is probable that you will be in danger, even in that case, a person can learn to, avoid that situation or be afraid of that situation. So, there, from the works of Joseph Liddox, Michael Davis, and Michael Fanslow, we now have a good understanding of neural circuits for both instinctive and learned fear in mammals. In particular, we know that both are centered on amygdala, which participates in the detection and evaluation of broad range of significant and potentially dangerous environmental stimuli, right? So we can look at the individual works of overall individual works of these three scientists who are actually relatively very young scientists, right? Joseph Liddox developed an interest in topic of emotion through his doctoral work with Michael Gazzaniga on split brain patients in mid 1970s, right? So that was when he has done his PhD. Because techniques for studying human brain were limited at that time, he turned to studies of rodents where the brain could be studied in detail. He chose to focus on a simple behavioral model, Pavlovian fear conditioning, right? This procedure allowed him to so it is a negative reward based conditioning probably, right? This procedure allowed him to follow the flow of information about the stimulus through brain as it comes to control behavioral responses by way of sensory pathways to the amygdala and gave rise to a notion of two sensory roads to amygdala, right? What were these two sensory roads? One was the lower road, which was a quick and dirty subcortical pathway for rapid activity behavioral responses to threats. The other one was a high road, which provided slower but highly processed cortical information, right? So if it went through the cortex, it was, highly processed information. But if it went through another pathway, it was quick and the response was rapid. Maybe it was not thought about as much, right? His work has shed light on how the brain detects and responds to threats and how memories about such experiences are formed and stored through cellular, synaptic, and molecular changes in amygdala, right? That was the work of Joseph Ledoux. Then comes Michael Davis, right? So I have kept this to show you that he's from Emory Department of Medicine. He has made significant contribution to neuroscience, particularly in the understanding of neural mechanisms of fear and anxiety. There are certain really nice book chapters also written by Michael Davis on this subject. His research has focused on amygdala again, particularly the central nucleus and the bed nucleus of the stria terminalis, which are critical for fear conditioning and anxiety. Davis studies have revealed the different parts of amygdala are involved in fear versus anxiety and has demonstrated the role of NMDA receptors in fear learning and extinction. Right? His research has also explored the impact of corticotropin releasing hormone on startle responses, linking it to anxiety as well as phobias. Davis' work has not only advanced our understanding of fear and anxiety, but also has implications for therapeutic approaches such as the use of D-cycloserine in psychotherapy to extinguish phobias of certain kinds, right? Then comes Michael Fanslow, right? So his research focuses on the neurobiology of fear, anxiety, and stress related behavior, very cohesive to the other two that we have just read about. He has developed the freezing assay of conditional fear, which is widely used in preclinical research of learning and memory, as well as fear and anxiety. So, Fanslow's lab has also developed animal models of PTSD, that is post traumatic stress disorder, and I am not sure if I expanded that correctly. That is why I'm taking that pause. Maybe you can check it. Capturing several major components of this disorder, Fanslow's work has significantly contributed to understanding the mechanisms of fear and anxiety and has also led to development of new research tools and models, right? Then again, we have talked about this a few times now that memory for learned fear in mammals involves the amygdala. This amygdala receives information about unconscious fear responses or emotional state directly, and the information about cognitive processing or of fear or feelings indirectly by means of connections from the cingulate cortex. Just a moment. Okay. In addition to its innate ability to respond to routine, natural threats, uh, yeah, the amygdala-based defense system is also able to learn quickly about new dangers, right? It can associate a new neutral or conditioned stimulus with a known threatening or unconditioned stimulus on a single paired exposure. And this learned fear can be remembered throughout the life, right? So, an example of this could be, for instance, if you ever saw a snake, which is, or a crocodile, for instance, right? You will remember getting feared or getting afraid of it, right? Even if it was, okay, say you are an animal lover. Say you saw a cobra biting a person, right? Like uh, putting its fangs in a person, or and the person immediately dying, right? You will be scared to death. Even if you are never going to come in contact with a cobra again, you will remember that memory throughout your life, right? Some things are like that. So that is what it means. So memory for learned fear in mammals involves this amygdala again, that is what we have been talking about. The long-term memory for learned fear in mammals also requires CREB that we have studied in the previous lecture also, that this is the molecular mechanism of memory storage in Aplysia and Drosophila also. In fact, in studies of amygdala, Alcino Silva, who you can see over here, and his colleagues have found that neurons in amygdala are recruited for long-term memory based on the basal levels of CREB expression, right? Neurons with large amounts of Krebs switch required for long-term memory are selectively recruited in fear learning. Then, indeed, the relative activity of Krebs at the time of learning determines whether a neuron is recruited. Conversely, if those neurons with a large amount of Krebs are selectively ablated after learning, the memory of fear is blocked altogether, right? So Pavlovian classical conditioning, if you remember, modifies the strength of synaptic transmission in the amygdala. In response to a tone, an extracellular electrophysiological signal proportional to excitatory synaptic response is recorded in the lateral nucleus in this case, and following pairing of the tone with a shock. Electrophysiological response is enhanced because of increased synaptic transmission between these neurons, right? Particularly in the amygdala. So, learned fear produces parallel and correlated behavior in electrophysiological changes. You can see this is a tone and this is a tone paired with a shock, right? So when there is training happening, there is a neutral response, right? So electrophysiological testing, when it is done from the lateral nucleus of the amygdala, the field enveloped potential looks something like this. An animal ordinarily ignores the neutral tone. The tone produces a small synaptic response in amygdala recorded by an extracellular field electrode over here. This field EPSP is generated by the small voltage drop between the recording electrode over here in the amygdala and a second electrode on the exterior of the brain as excitatory synaptic current enters the dendrites of a large population of amygdala neurons, right? But in case when a tone is presented immediately before a foot shock, right? So something is, the mouse is made to listen to something, but just after that particular stimulus ends, it is given shock, right? So whenever it will hear this sound, it will learn to fear the sound because it will know that next what is coming is probably a shock, right? Foot shock. So this is the kind of uh, this alternate brainwave pattern that you will see, or EPSP that you will see, right? So when the tone is presented immediately before a foot shock, animal learns to associate the tone with the shock. Now the tone alone will elicit what the shock previously elicited. Thus, the tone causes the mouse to freeze, an instinctive fear response altogether, right? After fear conditioning, the electrophysiological response in the lateral nucleus of the amygdala to tone is greater than the response prior to conditioning. Then these high frequency tetanic stimulation of either the direct or indirect pathway induces a long-term increase in synaptic response into these inputs. This change is a form of homosynaptic plasticity, which is known as long-term potentiation, as we have studied before as well. This long time synaptic change in amygdala may mediate fear conditioning altogether. The coronal brain slice from a mouse shows the position of the amygdala, as you can see over here. The enlargement shows three key input nuclei of amygdala, the lateral nuclei over here, basolateral over here, and basomedial over here, which together form the basolateral complex. These nuclei project into the central nucleus, which projects to the hypothalamus as well as the brainstem. The high frequency tetanic stimulation of the direct or indirect pathway to the lateral nucleus initiates long-term potentiation over here, right? So this is before the induction of long-term potentiation, and this happens after the induction of long-term potentiation. The drawing shows the position of the extracellular voltage recording electrode in the lateral nucleus and the position of two stimulating electrodes used to activate the either the direct pathway from the thalamus or indirect pathway via auditory cortex over here, right? So that is what you can see over here. Then if you look at this plot, it shows the amplitude of extracellular field potential, extracellular field EPSP, in response to stimulation of the indirect pathway as well as the cortical pathway during the time course of the experiment, that is on this x-axis, right? When a pathway is stimulated at a low frequency, which is once every 30 seconds, uh, then the field EPSP is stable. However, when five trains of high frequency tetanic stimulation are applied, the response is enhanced for a period of hours, right? The PK is active. The facilitation depends on PK and is compromised when PK inhibitor, which is KT 5720, is applied. The field EPSPs before and after induction of LTP are also shown over here. Then comes habit learning and memory, which requires striatum, right? It is again a part of that part near the hippocampus. So habits and routines are acquired gradually by repetition and result of a distinct form of implicit learning, right? So habit requires a repetitive kind of stimulation. So, for instance, if I read daily, I'll probably get into the habit of reading, right? If I eat a burger daily, I'll get into the habit of, eating junk daily. So, a habit is a stimulus response association, a behavior that is triggered simply by particular stimuli rather than by a desire for or a fear of some outcome, right? So many forms of habit learning depend on the four nuclei of basal ganglia that you can see over here. The striatum, uh, which includes, the caudate nucleus, the putamen over here. So caudate nucleus, putamen, and the nucleus accumbens, right? So all those three form the striatum. Then the globus pallidus over here, the substantia nigra over here, and the subthalamic nucleus over here, right? So these are the four nuclei of the basal ganglia. The striatum, the input nucleus, has three subdivisions in humans when we are talking about. These are caudate nucleus, putamen, and ventral striatum. Caudate nucleus is involved in certain forms of procedural learning, including stimulus response, associations, and some form of skill learning. It and the ventral striatum malfunction in a variety of diseases in which habit learning is disordered, including obsessive compulsive disorder as well as addiction. Striatum based implicit memory differs from the hippocampus based explicit memory in interesting ways, right? How does it differ? One of the differentiation points was given by Mark Packard and his colleagues, who demonstrated fundamental differences in neural structure underlying these two types of memory, implicit and explicit, right? So they tested both the types of memory using the same 8-arm maze, right? What they did was they took this 8-arm maze and they used the same maze for both kinds of memory. One is the explicit memory, the other is implicit memory, right? So in this explicit memory, what happens? In an explicit learning task, rat finds a food is available in at each end of the arm. Initially, a rat enters an arms at random, which with practice, the animal will learn to find all of the 8 morsels by entering each arm only once by following the path similar to the one that is shown by the dotted or dashed line over here, right? So a rat was placed in the maze daily and removed after it had collected food from every arm, and this was repeated for over several consecutive days. The rat's task was to minimize the wasted effort by remembering where it had already found food and to avoid revisiting those arms, and the rat had to shift its focus to unvisited arms, right? So it was that kind of a test. In performing this task, the animal has to acquire and use information about the single events. It must also remember the specific locations that it had visited on a given day, right? Just a moment. So this type of learning requires the hippocampus, the striatal region in the hippocampus, and if there is a lesion in the hippo in this region, then it is impaired. Also, if there is a damage to caudate nucleus, then it has no effect on this particular behavior of the explicit memory, right? But when it comes to implicit learning, the same kind of 8-arm maze is taken. In an implicit learning task, food is available only in 4 arms that are illuminated, right? The animal learns to visit each of these arms by associating this light with the food that is going to be provided. With this task, the animal needs to learn to visit 4 of the 8 arms of the maze that are identified by light at the entrance. Only these 4 arms contain a food reward. Over 2 weeks of training, the animals learn to revisit only the arms that were lit. The Wednesday task. So there was a wind shift task, which was the earlier one in the explicit memory, that once something is received in an arm, you have to shift to the next arm. If it has been received in that arm, you shift to the next arm. That was wind shift task, right? This was wind stay task, that once there is light in one arm, you have to stay over there, right? And go only when there is light in another arm, right? So it is disrupted by damage to caudate nucleus, but not by damage to the hippocampus. So this kind of implicit memory is probably more associated with caudate nucleus than hippocampus by itself. The two tasks are superficially similar because there is the similar kind of setup and similar kind of maze that is being used. But the Wednesday task requires the animals to learn about regularities that are constant from day one rather than to remember specific events on a given day. Right? As is the case with many forms of implicit memory, striatum based habit memory also requires CREB, right? What we had studied in the previous session. Animals with selective impairment of Krebs function in the striatum show impaired striatum dependent learning, right? then the learning induced changes in the structure of the brain contribute to biological basis of individuality, right? So there has to be some kind of a change or some kind of an alteration in the brain, whether it is or at the level of synapse or at some other level that will help the brain remember, right? So there has to be some tangible change, right? Although we can't go and look for that tangible change, but something that it should be present, right? So the process by which experience alters the somatosensory maps in the cortex is illustrated in an experiment in which adult monkeys were trained to use their middle three fingers at the expense of other fingers to obtain food. After several thousand trials of this behavior, the area of cortex devoted to middle fingers expanded greatly. Thus, practice may expand synaptic connections by strengthening the effectiveness of existing connections, right? So this is that example. So when the monkey was trained, before training, this was the brain area, right? This is how the brain area looked like. Post training, if you look at only these three middle fingers of this particular monkey, right? The area of the brain that is associated with only these three fingers, you can see how much it has increased post training, right? So, monkey was trained for 1 hour per day to perform a task that required repeated use of the tips of fingers 2, 3, and 4 occasionally. Occasionally 4 also, but generally 2 and 3. After training, the portion of the area 3B of the somatosensory cortex representing the tips of stimulated fingers, which is in the dark color over here, is substantially greater than the normal, which was measured 3 months prior to the training, right? Also, this similar kind of results were studied in adult humans, right? So, when human training was done, in which there was an acquisition of a motor skill in adulthood, right? So, this is the control, this is the trained person. A human subject was trained to do a rapid sequence of figure movements, which will improve the accuracy and speed after 3 weeks of daily training, right? So, if you look at this fMRI or functional magnetic resonance imaging scans of the brain, of the primary motor cortex of the brain. So we have still not come to the techniques, but fMRI, what it shows is the utilization of the oxygen by the brain. So it will be utilized by regions that are more active, right? So based on the local blood oxygenation level dependent signals show that after 3 weeks of training, the region activated in trained subjects, which is over here, orange, is larger than the region activated in control subjects who performed unlearned finger movements in the same hand, right? The change in cortical representation in trained subject persisted for several months, right? After the training had been performed. Then in this representation over here, you can see the cortical plasticity that is there in childhood. And x-axis over here is age of when a person began, their musical training. So the size of cortical representation of fifth finger of the left hand is greater in the string players than in non-musicians, right? So if you are playing a string-based instrument, you have a lot of movement of the fingers. So that is why it is used more. So there will be probably more area allocated to it. And this area allocation is not done at the time of birth or something, right? Eventually, how we train ourselves is how, different kinds of allocations are done in the brain, right? So the graph plots the dipole strength obtained from the magnetoencephalography, a measure of neural activity. The increase is most pronounced in musicians that begin musical training before the age of 13, right? You can see that the strength of the signal is highest in those players. And it is still higher than non-musicians if even after 13 years of age, but it is still not as high as if they had begun their musical training before the age of 13, right? So that is when the brain is also more plastic. There's more plasticity between synapses and all. Then the working memory depends on persistent neural activity in the prefrontal cortex, right? So we have understood what is working memory. It is basically the memory that is currently there in your brain, right? It is very similar to RAM. So for working memory to be present, there is something known as repetition that also needs to be done, or reverberatory, process needs to be involved. We'll read about that also. So, neuronal activity is measured while the animal is engaged in a delayed match to sample working memory task, right? In such tasks, the animal is initially shown an image or a sample and must retain the image in working memory for seconds to minutes after the initial animal is extinguished, or initial image is extinguished, or in the delay period. The monkeys are then showed a test image and must press a lever to indicate whether the test image matches the sample image, right? Then, if you look at it, the neurons in the prefrontal cortex are persistently, firing during the delay period, presumably contributing to the neural representation of the image in the working memory. Two major mechanisms may contribute to this persistent neural activity: the intrinsic property of neuronal membranes and the recurrent synaptic connectivity. Um, so these intrinsic membrane properties can generate persistent activity. Say this is our subject, a monkey, and this is the stimulus that it is being provided. So image is held, this image is currently held in the working memory. In some cortical neurons, a brief electrical stimulation can lead to persistent firing that lasts for seconds or even minutes after the end of the stimulus. Say after that, the screen has become blank, right? But you can see over here that sometimes there is persistent firing, as you can see in this three region over here, right? So intrinsic mechanisms of graded persistent activity. In the first one, brief depolarizing stimulus to a pyramidal neuron in the entorhinal cortex elicits a short burst of action potential followed by and after depolarization over here. Slightly longer stimulus elicits a longer burst of, as you can see over here, longer burst of spikes followed by large after depolarization. So after depolarization is also more as compared to the first one. And third is when the stimulus is further lengthened. The after depolarization is sufficient to trigger additional action potentials leading to persistent firing of tens of seconds, right? So, in this lower diagram, you can see it illustrates potential mechanism for this persistent firing, as you can see on the top. The influx of calcium ions through voltage-gated calcium channels (VGCC) during an action potential opens the calcium positive, calcium dipositive activated non-selective cation channels, or CAN channels. The resulting inward current through the CAN channels produces after depolarization over here that can lead to action potentials. The action potential further activate this VGCCs, the voltage gated calcium channels, perpetuating the cycle, and the recordings were obtained in the presence of carbachol, which activates the muscarinic acetylcholine receptors, a downstream signaling cascade that resembles the opening of uh, these calcium activated non-selective cation channels when intracellular calcium is also elevated, right? Okay, yeah, just a moment. So these network connections that are formed after a stimulus is, uh, given, right? It can sustain activity as well, right? The second kind of mechanism for sustained firing depends on recurrent synaptic connections within the neural circuit. So the first one was what? It was dependent on this graded persistent activity, right? The second one is going to be more dependent on the synaptic connections, right? The simplest case activities maintained by recurrent excitatory connections with the active population of neurons, right? Something like a feedback mechanism. The network can comprise either long-range connections between distinct regions of the brain or local circuits, or firing maintained through such chains, which is referred to as reverberatory activity. So it is a little more complex than simple positive feedback, but it is eventually going to kind of act like that, right? So we'll understand how this happens. So another circuit can sustain activity depends on reciprocal inhibitory synapses between two populations of neurons. When we visually see it, you will understand it better maybe. These neurons in both populations fire spontaneously at the basal level, and that is normally held in check by the reciprocal inhibitory synapses, right? So there is a normal basal level activity that is going on. But if there is a brief excitatory input into one of the populations of the neuron, it will transiently enhance their firing rate, which leads to increase in their inhibitory because they are working more. So because they were anyway inhibiting these neurons, now they will inhibit these neurons even more, right? Uh, After this, this decreases the inhibitory input onto the first population of the neurons. So there were two neurons, they were having a basal level activity, and with this basal level activity, they were inhibiting one another also, right? So then what will happen is this neuron had a more firing activity. Because of its increased activity, its inhibitory activity also increased, right? So this neuron population, its activity further decreased. So its inhibitory activity also further decreased, which further increased this first population's inhibitory activity, right? If you can follow that train. This decreases the inhibitory input onto the first population of neuron, further enhancing their rate of firing. This mechanism of positive feedback is termed as disinhibition. It can lead to firing of first population of neurons that outlasts the initial stimulus, right? Because this increased activity eventually is coming from decreased inhibitory activity from the second class of neurons. And you can see it over here as well. This is persistent reverberatory network activity, which is responsible for long-range synaptic interactions. So recurrent networks of synaptically coupled neurons can lead to persistent reverberatory activity. Um, Some network interactions occur between two widely separated populations of excitatory neurons in the distinct brain regions. As you can see, other excitatory networks are local, illustrated here by this reciprocally connected neighboring pyramidal neurons in the neocortex. In the third one, what I was saying is mentioned, right? Persistent activity can be generated through mutual inhibition. This example consists of two populations of neurons. One is group 1 neuron, the other is group 2 neuron. Within each population, neurons are reciprocally connected by excitatory synapses, right? However, the each population mutually inhibits each other. In this manner, an excitatory input to group 1, if there is an input to group 1, right, it leads to uh, the silencing of group 2 because inhibitory activity will also be increased, right? The loss of inhibitory input from group 2 eventually, and in turn, it will enhance the firing of group 1, right? So that is what we talked about. Although the relative importance of intrinsic activity versus network activity in the working memory remains uncertain, it is clear that the efficiency of working memory And persistent activity in prefrontal cortex neurons depends on the state of activation of the D1 type of dopamine receptors, right? So these receptors are coupled to G protein as GS or G stimulatory and the production of cyclic adenosine monophosphate or CAMP. So that response, a particular level of response of this dopamine or D1 receptor is required for the correct, activation. It was determined by Patricia Goldman Rasick and her colleagues, right, that has been mentioned over here. If working memory is most efficacious at the intermediate levels of D1 receptor activation, right? If it is less or more, in both cases, it can be pathological. Right, defects in dopaminergic regulation of working memory in prefrontal cortex are thought to contribute to the cognitive deficits which are associated with schizophrenia. Yeah. Then these explicit memory in mammals involves different forms of long-term potentiation within the hippocampus. Unlike working memory, long-term storage of information by hippocampus is not thought to depend on persistent neural firing, but rather to involve long-lasting changes in the strength of synaptic connections itself. Right? So this is a hippocampal circuit, which is important for the declarative memory. Uh, the information over here arise in hippocampus from either the entorhinal cortex through the perforant pathways which provide both direct and indirect input to CA1 region. This is CA1 region in the hippocampus. It contains pyramidal neurons and the major output neurons of the hippocampus. So you can see there are two kinds of inputs. One is direct, the other is indirect. The indirect one is trisynaptic, right? In the indirect trisynaptic pathway, in the layer 2 of entorhinal cortex over here, uh, uh, they send their axons through the perforant path to make excitatory synapses onto the granule cells over here. Of the dentate gyrus, the granule cells project through the mossy fiber pathway to make excitatory synapses with pyramidal cells in the area CA3 of the hippocampus. CA3 cells excite the pyramidal cells over here in the CA1 region by means of the Schaffer collateral pathway and the direct pathway neuron in layer 3 of the entorhinal cortex project directly through the perforant path to make excitatory synapse on the distal dendrites of the CA1 pyramidal neurons without intervening synapses, right? Different neural mechanisms underlying long-term potentiation at each of the three synapses in the trisynaptic pathway in the hippocampus. Long-term potentiation is present at synapses throughout the hippocampus but depends to different degrees on the activation of NMDA type glutamate receptors, right? So I think these are the final few figures that I have kept for today's the discussion. The first one shows tetanic stimulation of Schaffer collateral pathway, which induces LTP at the synapses between presynaptic terminals of CA3 pyramidal neurons and the postsynaptic CA1 over here pyramidal neurons. The graph plots the size of extracellular field EPSP expressed as percentage of uh, initial receptors of CA1 percent of initial baseline of FEPSP prior to induction of the long-term potentiation. At these synapses, long-term potentiation requires activation of NMDA receptors in CA1 neurons as it is completely blocked when the tetanus is delivered in the presence of NMDA receptor antagonist AP1, APV, right? So you can see that in this case, this response completely is missing because NMDA receptor is basically blocked, right? So that might be the crucial receptor in this path. Then the tetanic stimulation of direct pathway from entorhinal cortex to CA1 over here generates LTP of EPSP that depends partially on the activity of NMDA receptors and partially on the activity of L-type voltage gated this one CA2 plus channels. It is therefore only partially blocked by APV and addition of APP and natridipine, which is a dihydropyridine that blocks L-type channels, is needed to fully inhibit the long-term potentiation, as you can see over here, right? Then comes the third pathway, which is the mossy fiber pathway. The tetanic stimulation of mossy fiber pathway induces the long-term potentiation at synapses with the pyramidal cells in the CA3 region. In this experiment, the excitatory postsynaptic current was measured under voltage clamp conditions. This LTP does not require activation of NMDA receptors and is also not blocked by APV, right? It requires activation of protein kinase A, so it is blocked by kinase inhibitor H89. So, as I said, that was the end of this particular discussion. There is a very short portion of this learning and memory that is still left, and I think I'll take one entire more session, that short part that is from the book Candle that is left, and briefly I'll also go about this discussion of learning and memory in terms of cognition, right? So there is that other book that I had suggested, Student's Guide to Cognitive Neuroscience by Jamie Ward, that like its perspective on learning and memory, we'll briefly go through. So 1 or 1.5 sessions, we'll spend more on this anyway, because you are learning about neuroscientist histories. That portion, it is detailed enough in your main lecture that sir is taking, so I don't feel we need to elaborate more on that over here. If you have any question, if you want me to explain any particular experiment that they had done, that I can do over here, you can write to me for that, you can tell me over here also. But after I'm done with this learning and memory part, what I have personally planned is we will go through the different methods to study brain, right? So, the different techniques that I used, right? Clinically, majorly. Probably I can keep something that is used in the labs also that we do, right? To look at different states of the brain, X, Y, O mostly. So, yeah, that is going to be it. Uh, so if there is anything I can help you with, please do tell me, or you can write to me as well. I'll upload the recorded lectures also. Um, if not, thank you so much for attending the session. Good night to all of you.